Aging, A to Z Futurism. Aging is the process of becoming older. It refers mostly to humans, many other animals, and fungi, whereas for example, bacteria, perennial plants and some simple animals are potentially biologically immortal. Aging can refer to single cells within an organism which have ceased dividing. In humans, aging represents the accumulation of changes in a human being over time and can include physical, psychological, and social changes. Reaction time, for example, may slow with age, while memories and general knowledge typically increase, and aging increases the risk of human diseases such as cancer, Alzheimer's disease, diabetes, cardiovascular disease, stroke and many more. There are three well-known and researched categories or so-called types of aging: biological aging, psychological aging, social aging. The descriptions in this video of aging and the treatments for this condition is only focused on biological aging. It does not refer to psychological or social aging. Current statistics suggest that of the roughly 150,000 people who die each day across the globe, about 2/3 die from age-related causes. The largest causes of death are in highest rates of death to lowest as follows: heart disease, cancer, respiratory disease, strokes, Alzheimer's disease, diabetes, influenza, pneumonia, kidney disease and Parkinson's disease. Solutions to aging includes 1. Physiological, 2. Biochemical, 3. Genetic. Current theories of aging indicate that it is a physiological problem that is linked to specific genes, cell loss, cell atrophy, telomere dysfunction, mutation, mitochondria dysfunction, cell accumulation, intracellular junk and extracellular junk. SENS, or Strategies for Engineered Negligible Senescence, is a framework of regenerative medicine therapies aiming to extend healthy human life by repairing age-related damage at a cellular and molecular level. Pioneered by Aubrey de Grey, SENS proposes that aging is a collection of specific damages that can be addressed through repeated interventions, preventing the decline into age-associated diseases and achieving a state of negligible senescence. For more information, refer to my videos on gerontology, immortality, life extension, indefinite life extension. The SENS Foundation identifies relevant research and ways to combat aging. This includes 7 key research areas such as gene therapy, pharmaceuticals, gene editing, vaccines, suicide virus, stem cells, and biopharmaceuticals. The central theme to the physiological solution to aging is blood, and in particular the blood of young people. There are experiments that were performed in mice where the young blood of mice into old mouses lead to life extension. This is said to be the holy grail of proteins and so-called bloody pills. Parabiosis in the context of aging experimentation. Heterochronic parabiosis is the surgical circulatory fusion of mice of different ages and allows mechanistic interrogation of two processes simultaneously, the effect of aged blood and systemic milieu on the young parabiont, and vice versa, the effect of young blood and systemic milieu on the aged parabiont. Emerging literature emphasizes the utility of the heterochronic model to reveal rejuvenated mechanisms originating from the youthful circulatory environment and, conversely, proboronic mechanisms originating from the aged circulatory environment. Heterochronic pairing must be contextualized with isochronic control pairs, comprised of same-age mice, young and old, as well as young and old non-paired mice, which enable experimental standardization of age and procedure-dependent outcomes. Stem cells and regeneration. Scientists have made a breakthrough discovery of a key protein that supports the production of healthy blood cells throughout life by regulating the body's inflammatory response. The study is the first to reveal a protein that has a crucial role in protecting the blood stem cells. The stem cells continually produce all blood and immune cells needed in the body from premature aging. The discovery shows that the protein, known as YTHDF2, protects these stem cells from damage when the body is fighting an infection. This protein allows stem cells to continue functioning correctly throughout life. Scientists were bewildered by the stem cells known as hematopoietic stem cells and how they protect themselves from burnout whilst ramping up the production of immune cells to combat an infection. Previous studies found that the YTHDF2 protein has an important role in the development of healthy blood cells. This is a process known as hematopoiesis. Curiously, Scientists have studied starfish and revealed that there is a concept called stem cell regeneration. The starfish in the snail fur can actually achieve stem cell regeneration. If one were to look for enhancement of the human healing, it is through studying starfishes that we might one day be able to heal faster and efficiently. Notably, if a starfish loses one of its arms, it can grow it back just the same. This is due to the starfish's unique cells, which can differentiate themselves from skin or muscle cells back into stem cells. The purpose of regeneration on a physiological level is to rejuvenate humans, and this is often related to the Yamanaka factors. The Yamanaka factors October 3/4, SOX2, KLF4, CMYC are a group of protein transcription factors that play a vital role in the creation of induced pluripotent stem cells, cells that have the ability to become any cell in the body, often called iPSCs. They control how DNA is copied for translation into other proteins. The essential question is if Yamanaka factors can reverse aging. A separate team followed a similar strategy to reverse aging-like changes in genetically engineered mice. In both cases, the Yamanaka factors appear to have restored part of the animal's epigenome, chemical modifications on DNA and proteins that help regulate gene activity, to a more youthful state. The simple answer is yes, rejuvenation and a return to youth is possible, but in humans it is far more complicated. As suggested by scientists, in the future there is intention to store stem cells for later use. Utilize the nanog gene, nanog homeobox, as a protein-coding gene. Stem cells could be the key to different new health-related technology, gene therapy, and improving longevity in the future generations. More research into multipotent, totipotent, and pluripotent stem cells is most certainly likely to occur. Hidden human genes. Gene editing can regeneration of skin and healing of physical wounds. It is thought that the right enhancements can make this a reality. MG53 fixes the cell and tissue damage that occurs during everyday living. Even simple actions, like walking or typing, will cause injuries to the body. Usually this isn't a problem because MG53 can make repairs before there's any serious harm. More importantly, healing DNA is important. Most damage to DNA is repaired by removal of the damaged bases followed by resynthesis of the excised region. Some lesions in DNA, however, can be repaired by direct reversal of the damage, which may be a more efficient way of dealing with specific types of DNA damage that occur frequently. Homeotic genes are master regulator genes that direct the development of particular body segments or structures. Homeotic genes are the master control genes. They regulate groups of genes that determine what body parts will develop in which locations. Master control genes could be the key agelessness and youth. A curious film about agelessness is Man from Earth. It is a 2007 American science fiction drama film directed by Richard Schenkman. Regeneration is tissue engineering through gene modification, editing, whilst rejuvenation is repairing DNA and other aspects of the body. The so-called hidden human genes are Lin28A gene and IMP-1. Researchers suggest turning this gene off can be risky, but in some cases may prove to be beneficial against potential cancers and aging. The gene Lin28A encodes a Lin28 family RNA binding protein that acts as a post-transcriptional regulator of genes involved in developmental timing and self-renewal in embryonic stem cells. Lin28 enhances tissue repair by reprogramming cellular metabolism. Thus cellular reprogramming and epigenetic rejuvenation is an ongoing research in the field of gerontology. Cellular reprogramming has demonstrated potential not only in regenerative medicine, but also in the aging field through the amelioration of both physiological and cellular aging. Hallmarks. Reprogramming-induced rejuvenation, RIR, is a better term, capturing the nature of the utilized process and final aim of the interventions. RIR has shown promise as a treatment to safely reverse aging whilst retaining the ability to revert to or maintain original cell identity, both in vivo and in vitro. Tissue engineering. Tissue engineering is associated with applications that repair or replace portions of or whole tissues. The term regenerative medicine is often used synonymously with tissue engineering, although those involved in regenerative medicine place more emphasis on the use of stem cells or progenitor cells to produce tissues. The goal of tissue engineering is to assemble functional constructs that restore, maintain, or improve damaged tissues or whole organs. Lab-grown organs usually involves 1. Refurbishing. 2. Scaffolding. 3. Bioprinting. The examples of artificial tissue engineering are in vitro meat, bio-artificial liver device, artificial pancreas, artificial bladders, cartilage, scaffold-free cartilage, bio-artificial heart, tissue-engineered blood vessels, artificial skin, artificial bone marrow, tissue-engineered bone, laboratory-grown penis, oral mucosa. Various companies sells a liver on a chip, with prices for the liver chip starting at about $22,000. Using this technology is faster and far more cost-effective than animal research. Daniel Dennett's Where Am I? Is a famous philosophical science fiction story where Dennett gets his brain removed. He then asks himself, why is he conscious in his body and not in his brain? This causes multiple explanations and possible answers to arise. Dennett claims that our brains hold only a few salient details about the world, and that this is the only reason we are able to function at all. Therefore, we do not store elaborate pictures in short-term memory, as this is not necessary and would consume valuable computing power. Brain transplants films are 1. Transfer 2011. An aging rich couple pay to have their minds transferred into younger bodies, but they find their arrangement becoming unpredictable when they learn that their hosts can reclaim their bodies for 4 hours each night. 2. Self-less 2015. A scientist transfers the mind of a terminally ill billionaire into an artificially grown body, but the result is not quite what he expected. 3. Criminal 2016. CIA agent Bill Pope is on a mission to save a hacker from a dreaded criminal. However, when the agent is mysteriously killed, his memories are grafted into Jericho Stewart, an ex-convict. Biological aging of the brain and neurological aging of the brain is the most difficult part of the physiological solution to aging. This is because the brain is thought to be the most complex organ in the entire universe. Additionally, consciousness may also be outside the brain, which lead to a better understanding of how it connects to the human biofield in the universe. Neuroscience today says consciousness is generated by and localized in the brain because it emerges from brain activity. Alternatively, various researchers propose that consciousness may not originate in the brain, although some aspects of human perception of consciousness may be dependent on the brain. The genetic solution to aging. Customized genes and editing our genes in vivo may be the best solution to aging as suggested by medical Harvard School. Can we customize genes? As we age, genes that maintain cellular health and vitality are downregulated, and at the same time, genes that promote disease and senescence become overexpressed. Once physicians are able to precisely program or edit DNA genes, then youthful health may be systemically restored. This is the often known as rejuvenation and associated with the DNA damage theory of aging. Life extension slows aging, whilst rejuvenation is the practical reversal of the aging process. DNA damage contributes to aging via cell autonomous events such as causing apoptosis, which depletes functional cells such as neurons, and via cell non-autonomous mechanisms, such as triggering senescence, which can negatively impact the function of neighboring undamaged cells. As an example, a study showed that centenarians of 100 to 107 years of age had higher levels of 2-DNA repair enzymes, PARP1 and Cu70, than general population old individuals of 69 to 75 years of age. Their analysis supported the hypothesis that improved DNA repair leads to longer lifespan. Moreover, a gene called GATA6, GATA-binding protein 6, regulates aging of human mesenchymal stem stromal cells (MSCs), according to new research from the University of Wisconsin-Madison. The research, published in the journal Current Biology, discovered that individuals with one form of a gene called MC1R look 2 years older than those with a different form. The klotho gene owes its name to this goddess because of its scientific association with anti-aging properties. By the age of 40, the human body's klotho levels naturally decrease. Klotho also regulates a protein called WNT5A that's been shown to advance the spread of cancerous cells and treatment resistance. Gene therapy. A significant part of aging is controlled by so-called master genes. If the activity of such genes can now be intentionally controlled through the means of CRISPR tools, then slowing down or even reversing the aging process is theoretically possible. One study used a gene therapy, introducing 3 genes, known as Yamanaka factors, that are particularly active in embryonic cells. According to the authors, the remaining life expectancy of the mice whose age was equivalent to about 77 years in humans was doubled, with a 7% increase in absolute bioluminescence. Trees glowing in the dark. Trees and plants, high-yield crops, reversing and slowing the down-aging process. Currently, a natural version of CRISPR has been adapted by scientists to enable the reprogramming of cellular DNA to rid cells of unfavorable genetic changes. Once perfected, old cells may be rejuvenated and never age again. However, there are several issues with CRISPR as listed below. Issues with CRISPR. 1. Eugenics, we all become the same. 2. Tribalism, we all become different. 3. Bioterrorism, we become ill and sick. 4. Playing God, we become creators. For further queries about genes, I'd highly recommend the term and definition abolitionism, where I systematically list all genes relevant to academic research and their purposes. See links in the description or simply visit the A to Z Futurism playlist. In addition, I'd recommend my videos on the terms gerontology, immortality, and life extension. You may have realized I did not speak of digital immortality or cyborgism. For more on these topics, see my video Indefinite Life Extension and Immortality, where I explain the difference between biological immortality or digital immortality. Thank you for watching this video, and I hope today we have learned the basics of some genetics, aging and concepts or theories related to these topics. Thank you again and have a great day.